Hello everyone. We are today in the fourth class of cytokinin and we are going to understand the cytokinin signaling in plants. Before we understood about the biosynthesis of cytokinin, the history of cytokinin discovery, and even we have completed the role of cytokinin in agriculture. That was an important lecture. If you have missed, you just go to the channel and visit and listen to the lecture. I would suggest if question are there, please raise question and I will make separate videos for each question even. Okay, so let's start the fourth important section of cytokinin, that is the cytokinin in plants. Okay, so we have this image where we are going to understand the whole mechanism of signaling in plant cell. Okay, so let me explain it in basic language so that you people can understand the whole mechanism. Okay, so whenever one cell is there, in the membrane there must be a receptor. For signal transduction, we need one receptor in the cell membrane. That receptor will receive the signal and it will propagate in the through the cell or cytoplasm, and then it will enter to the nucleus and necessary changes can be made in the DNA. Okay, this is the process. So this whole process is called as signal transduction, or in other language we can call it as a signaling. Okay, so here in cytokinin signaling, one receptor is there. We call it as CRE1, and there are a number of homologs of it. Some are AHK2 and other AHK3. Okay, so these receptors are having three type of domain. Okay, number one is chase domain, which is placed in the membrane. Second one is your histidine kinase domain. Okay, this is the histidine kinase domain. Third is the receptor domain. Third part is the receptor domain. So basically this, it is having three parts. One is the chase domain, second is your histidine kinase domain, third is your receiver domain. Okay, so three parts are there. Three parts are there. So the what is happening, one histidine kinase domain is there. What is happening, whenever cytokinin is attached to this chase domain is making changes in the whole cytoplasm. Okay, or signal transduction process is going on. Okay, so here in this histidine kinase domain, it will break ATP and release phosphate group, and that phosphate group will attach to this histidine kinase domain. And when signaling is going on, this phosphate group will be transferred to your receiver domain, and receiver domain will again transfer it to the AHP. Okay, we will understand what is AHP. Now this AHP will act as a transcription factor. What is a transcription factor? Transcription factors are molecules or biomolecules, mostly proteins. They can enter to the nucleus. Such protein which are entering to the nucleus and changing in making changes in your DNA, we call it as a transcription factor. So this AHP or phosphorylated AHP, we can say, is acting as a transcription factor, and it is entering to the nucleus. Okay, and in nucleus it is getting attached to type BARR, or the receiver domain of type BARR is phosphorylated for further changes. and type B ARR is what it is doing. It is changing in DNA to form mRNA, which will be attached to the type B ARR, type A ARR. Type A ARR will phosphorylated and it will make other changes which will create cytokinin response in the cell. Okay, and some unknown factors are there which through which type B ARR is basically showing some feedback response. So that part is basically unknown. Okay, so let's see whole process in text. Okay, in Arabidopsis, the binding of cytokinin to its receptor involves a complex process. It is basically a complex process. Cytokinin typically binds to CRE1 or its homologues AHK2 and AHK3, which are believed to exist as dimer. The binding specially occurs at the extracellular segment of the receptor known as chase domain. Essentially, cytokinin engages with chase domain present in CRE1, AHK2, AHK3, suggesting that these three hybrid sensors kinase are potentially receptor for cytokinin arabidosis. This interaction plays a crucial role in cytokinin signaling pathway within the plant cell. Okay, upon cytokinin binding to these receptor, it triggers the activation of their histidine kinase activity. As a result, a phosphate group is transferred to an aspartate residue located on the fused receiver domain of their receptor protein. The phosphorylation event is the key step in the इंटरसेलुलर सिग्नलिंग कास्केड. ओके, सो बेसिकली क्या है, जितने सॉरी जो जो फॉस्फोराइलेशन हो रहा है और the process of phosphorylation is, if we see, is repeated. Okay, first this histidine kinase domain is being phosphorylated here, then receiver domain is being phosphorylated here in CRE1, then AHP is being phosphorylated, then AHP also is entering into the nucleus and phosphorylating type B ARR and type A ARR. Okay, so this way, if we see whole the signaling process, the key of the process is this phosphorylation event. Okay, following the phosphorylation of aspartate residue on the receptor, the phosphate group is subsequently transferred to a conserved histidine residue found within AHP. Now, what is AHP? Arabidopsis histidine phosphotransfer protein. As I told you, these transcription factors are mostly proteins. Here it is also a protein, Arabidopsis histidine phosphotransfer protein. This transfer of phosphate from the receptor to AHP protein is a pivotal step in cytokinin signaling pathway, facilitating the relay of signal further downstream for cellular responses and gene regulation in the plants. Upon phosphorylation, the AHP protein undergoes translocation into the nucleus. Okay, within the nucleus, it executes transfer of phosphate group E to your ARR. Okay, aspartate residue situated within the receiver domain of type B ARR. ARR meaning Arabidopsis response regulator. Okay, Arabidopsis response regulator. There are two type of ARR here. One is type A ARR, another is type B ARR. So both are getting, or both are being phosphorylated, but if type A is the key for the response, if type B is also phosphorylated, even phosphorylated, but it is basically activating type A. Okay, and type A can directly be phosphorylated for activation. Okay, so basically, the transfer of phosphate from AHP to aspartate residue of type B ARR is a crucial step in cytokinin signaling pathway, enabling the activation of type B ARR and subsequent regulation of gene expression to orchestrate plant response to cytokinin stimuli. Now, type B ARR is phosphorylated. This activation even stimulates its output domain. Okay, this output domain leading to the initiation of transcription of genes encoding type A ARR. That means type B ARR is basically producing type A ARR. Okay, this activation cascade sets off a regulatory cycle in cytokinin signaling pathway, influencing the expression of genes involved in various response and fine-tuning the plant's reaction to cytokinin signal. So ultimately, this type A ARR is showing your cytokinin signaling or cytokinin response. Okay, the AHP protein are also likely to responsible for phosphorylating the type A error directly. Okay, this phosphorylation process represents another layer of regulation within the cytokinin signaling pathway, potentially influencing the activity and function on type A error in cellular responses to cytokinin signals. Phosphorylated type A error engages with diverse effectors, facilitating the modulation of cell function that align cytokinin signaling. These interactions play a crucial role in orchestrating the array of cellular responses designated as cytokinin responses within the model, enabling the plant to enact appropriate changes in response to cytokinin stimuli. Okay, so this is the whole signaling process. Let me explain you or brief you in short so that you will not forget this concept. So as I told you, the phosphorylation is the key process. Okay, when one cytokinin molecule is being attached to the receptor, and receptor which is having three domain, one is chase domain, one is histidine kinase domain, third is your receiver domain. So cytokinin will never enter into the cell, but it will just attach to the receptor, and after attached with the receptor, whole changes inside the cytoplasm will take place. Okay, and whole, what is the main process? Basically the phosphorylation. Okay, first the phosphate group will attach to the histidine kinase domain, where from where it is coming, it is coming from breakdown of ATP. Okay, now histidine kinase will transfer the protein and phosphorylate the receiver domain of the receptor. Okay, this receptor is now phosphorylated or activated. It will now activate your AHP protein, which will act as a transcription factor. Now it will enter into the nucleus through nuclear pore, and this AHP will phosphorylate your type B ARR and also directly type A ARR. Let's see what is happening when type B ARR is being phosphorylated. Okay, so type B ARR, if is phosphorylated, it is basically changing the DNA or activating the transcription of type A ARR, and type A ARR is being produced or being formed, and type A ARR will now phosphorylated and will cause responses, cytokinin responses. So this is all about your cytokinin signaling. I hope you people have understood the concept. If you are not getting it, comment me. I will take another class and simplify whole the process as easy as possible. Okay, thank you. Please show some love and respect to this channel through liking, subscribing it, and sharing it more and more. Thank you so much.